Hey, Biotechnology students, Mr. Yance again. Let's finish up notes today. So we're going to finish up this biotechnology section. Mainly, it's a good topic. We're just going to kind of wrap it all up here and show you how, we've been going through the different tools. Talked about some of the controversy with biotechnology, with cloning and stem cell research, and are we always using biotechnology for the good and useful products? That was what the definition says, but we don't always follow that. So there are some good things going on in biotechnology, and that's what I kind of want to throw out there today. Diagnosis of disease is a big one. We're able to, I showed you that microassay and how we can identify alleles and viral DNA, and was big in diagnosing, figuring out COVID-19, which is what we've been struggling with, this year. Gene therapy. We kind of pumped our brakes on that a little bit because that's our ultimate goal of biotechnology, is to take this to the next level and cure diseases and cure illnesses. And we do that somewhat, but not to the extent we'd like to. We had to pump our brakes on this because we had some people die in some gene therapy studies, and we decided we maybe need to look at this a little bit more. We don't know if they died of the altering of the afflicted gene, or they died of just the disease itself. So we're still trying to figure that out. Production of pharmaceuticals, COVID-19. Hey, what's up? Okay, so we're talking about forensic applications. We did a little bit of DNA profiling with our lab that we did over there with gel electrophoresis. That's another big part of biotechnology. I'm going to talk more about that in a little bit. And this is a big one too for the future, is maybe environmental cleanup. Can we inject a gene into bacteria, things like that, and clean up oil spills, eat up the petroleum products, whatever. And a highly controversial one I'll end notes with is GMOs. So genetically modified crops and foods and things like that. So let's kind of look at gene therapy for just a second. So here's what we're talking about. We got a clone gene. We put it, let's say that someone has a bone marrow cancer. Then why can't we take a normal healthy gene, put it in a retrovirus, which doesn't mean it's not harmful to us. It goes in there, it duplicates that gene. You let it grow, culture it, stick it back into the bone marrow cell from the patient. It puts it takes this gene, puts it into the chromosomes, the DNA. Now we can inject it back into the person. That's the idea. Okay, so that's gene therapy, and that's what we're looking to do, is not treat genetic diseases, but maybe someday cure them, right? And then we have this thing called farm animals, not farm, F-A-R-M, farm, P-H. Here's a goat. We put the human protein in this goat, and when she secretes and produces milk, instead of using it for consumption like we normally use milk, we use it for medical use, and we can make drugs and treatments and things from that milk. Kind of crazy, but another method that we can use. Okay, DNA fingerprinting. I want to talk a little bit about it. DNA fingerprinting has changed a lot in recent history. It dates back to 1984, where it was the first time we started to use DNA to identify people. And since then, it's transformed CSI, criminal scenes investigations, paternity cases. You watch that on show, am I the mom or dad? Usually the mom we know, but the dad's the one always is in question, it seems like. Now, understand a couple things. So if you're looking at DNA and gel electrophoresis like we did in the lab, we have a crime scene evidence, and then we have a suspect 1, suspect 2, and a victim. We rule out the victim, and you can see the suspect 2, in this case, same way ours worked, matches the crime scene. He was at the crime scene 1. Okay, so we use that a lot. Now we have a new technique we're using now. It's a little more accurate. I'll show you that in a second. Paternity is different, because paternity, you're not going to get a child that is an exact match to a parent, because you're getting 23 chromosomes from mom and 23 from dad, right? So that is a little bit different, but you can see this is an interesting one. We got mom and dad. Child 1 and 3 are definitely children of both parents because they have markers from both parents showing that, yeah, they are their children. Child 2 is interesting. It's got moms, but she does not have any of dads. So dad is not her father. And child 4, poor guy, he doesn't have any of them. So he must be adopted. So that's what we use for paternity cases. So when they open up that big envelope and those crazy shows on TV, it says, oh, the results are in and you are the dad or whatever. That's how we do that. Okay. Now, here's the old stuff. We still use this. It's still effective. It's still accurate. The problem is E up here on the screen. It takes a pretty good sample size. RFLP, restriction fragment length polymorphism, is a fancy word for that. We take restriction enzymes. We talked about that, and we cut it. And we get those sticky ends. I showed you all that. And then you run that through a gel, rectophoresis. And you can do a couple things with that. You can analyze it, see if someone has a disease, or you can use it for criminal investigation, see if someone was at a crime scene. Okay. Problem is, it's a little outdated for profiling for crimes and things like that because you have to have a quarter-size sample, which is quite a bit, and sometimes people don't leave that much behind at a crime scene. So we've kind of shifted over to, and here's a good example of using it for disease diagnosis. There's someone has a normal allele. Here's someone with sickle cell, and they don't produce the same blood cells that a normal one would. So we still use it a lot for disease diagnosis, and we still use it for some crime scene stuff. But here's the new stuff. I'm going to skip this video. I'll post it online. I'll do that today, and you're welcome to look at that if you'd like, especially you virtual students. Now we use something called STR, which stands for short tandem repeats. We use a non-coding part of DNA, which means it doesn't code for a trait. And we know that people have these non-coding DNA regions, about 2 to 5 base lengths, and they repeat. So what you can look at these repeats, and they're identifiers. So all of us have different sequences that repeat, and it identifies us as an individual. Okay. The nice thing with this is you only need 20 cells, and you can get that from skin, saliva, semen, anything left behind at a crime scene. Doesn't take much to get 20 cells. Okay, maybe hard to get a quarter-size sample. 20 cells is nothing. Okay, and you run it through, and you can see that here is 22 chromosomes that are body chromosomes in the male and female, which in this case would be the male XY. And you can see these repeats, and we can identify these regions, okay? So you run it through an analysis, and you can see here is an analysis of a person that has, here's their repeats, okay? So you have a person up here in blue, and then green, and yellow, and then red, okay? So here's kind of crazy stuff. So this guy was in prison for 17 years. And they did this new technique. They convicted him, put him away for life in prison. He had raped a lady and killed her, and they convicted him, and he was in prison for 17 years. And then they did this new technique, and look what happened. Here's what was left behind the crime scene. He doesn't match any of these markers, but yet this guy named Kenneth Tinsley matches all of them. So guess what? Earl got set free, and Kenneth got stuck in prison the rest of his life. So it does exonerate. It does sometimes convict people. It works both ways. So we're doing a lot. There's a group out there that's looking at cold cases like this and using STR and seeing if the people we've convicted for crimes were really the people that did the crime. I mean, they all say they're innocent. I get that. But in some cases, they truly were. There's a great example of that. Okay. And lastly, genetically modified crops, organisms are altered. You insert a DNA into a genome. For example, if I wanted to, well, let's put one up here. Okay. This is probably the most famous one that we started with. There's cotton and corn that is very susceptible to caterpillars. And caterpillars get in there, they eat the leaves, shut down photosynthesis, and the crop dies, and farmers are losing millions of dollars because of crop damage from caterpillars. Okay, so scientists figured out that there's a bacteria called Bacillus thuringiensis. There you go. commonly occurs in soil bacteria. It's a common bacteria. And they found that this Bt gene in this bacteria, if a caterpillar eats it, it turns toxic in their digestive system and they kills the caterpillar. So we've taken that Bt gene out of the bacteria, inserted it into cotton and corn, and then as that crop grows and those leaves grow, when the caterpillar eats the first leaf, it kills the caterpillar, and it's saved a lot of crops. And here's the thing. People get all shook up. I don't want to eat GMOs. That's going to be harmful to me. It does nothing to people. It is a harmless gene. It's in the leaves. It's not in the corn that we're actually consuming. And I think that's what we have to understand. A lot of these GMOs are engineered, here's the rest of that, so that, yeah, they take care of a particular thing, drought, disease-resistant caterpillars, nematodes, whatever, but they're not going to get into the food supply and make us sick. We've been modifying foods for a long time. You know, I plant some GMO corn and soybeans for my uncle, about 300 acres, and they're perfectly safe. They have geneticists, they have researchers that test these things. So be careful what you read on social media. You can make your own decisions on what you're going to do, but we've been modifying these crops for a long time, and nature has been genetically modifying them for a long time. So you just got to keep that in mind. Our food's safe. It's all right to consume. All right. Okay, that's it. So that's it for notes. Hopefully that makes sense. If you got any questions, let me know. We'll do a review game. We'll take our test next week. All right. Hey, have a great day, and we'll see you later. See you. Bye.